Today, I'll be sharing with you my project in which we looked at the effects of perinatal iron deficiency on neonatal cardiac maturation. Iron deficiency is the most common nutritional deficiency in the world, and pregnant women are among some of the most vulnerable groups. Iron deficiency during pregnancy can alter the developmental trajectories of the offspring and thereby predispose them to cardiovascular diseases later in adulthood. Previously, our lab has shown that prenatal iron deficiency causes both a sex and organ-specific pattern of hypoxia, mitochondrial dysfunction, and oxidative stress in the fetuses of iron-deficient dams. However, one organ that has not been studied in the context of perinatal iron deficiency is the developing heart. The neonatal heart is highly metabolically active and are also undergoing a number of important developmental changes during this period, and therefore it might be particularly vulnerable to the effects of perinatal iron deficiency. Most recently, our lab has found evidence of cardiac dysfunction in the hearts of neonatal iron deficient rats using echocardiography. However, the mechanisms behind these changes are still currently unknown. Therefore, the objectives of my project is to determine if perinatal iron deficiency disrupts cardiomyocyte maturation postnatally, and also to see if these changes persist throughout the neonatal period. We used a maternal iron restriction model in which female sprawled dolly rats were fed with either control or iron deficient diet both 2 weeks before and during their pregnancy. After birth, all dams and pups were switched on to a standard rodent chow, and heart samples were collected from male and female pups on postnatal days 1, 4, 14, and 28. These heart samples were then used in Western blots to look at markers of cardiomyocyte maturation or embedded and sectioned for histological analysis using Mason's trichrome staining. First, let's look at the birth outcomes. As expected, male and female iron deficient pups had decreased levels of hemoglobin on postnatal days 1 and 14, but had recovered by day 28 after they were weaned and started eating their own chow. The iron deficient pups were also growth restricted past day 28 and also had increased relative heart rates on days 1 and 14. We then used Western blots to look at the ratio of alpha to beta myosin heavy chain. This ratio in rodents increases after birth and is a marker of cardiomyocyte maturation. We see that on days 14 and 28, the iron deficient pups had an overall lower ratio of alpha to beta isomers, and this may indicate that there is some disruptive maturation happening with their cardiomyocytes. There is a significant interaction, in fact, between sex and diet noted on day 28. However, the significance of this is still currently unclear. Next, we compared various histological measurements using neonatal heart sections stained with Mason's trichrome staining, seen here. However, none of these comparisons, such as the wall thicknesses and ventricular diameters, were significant. After that, we compared cardiomyocyte cross-sectional areas in these same heart sections, also stained with Mason's trichrome stains, and some of the selected data is shown here. In panels A, B, and C, we compared the cross-sectional areas between iron deficient and control pups, and we see that on postnatal day 28, there is a significant overall increase in the left ventricle and right ventricle cell area in the iron deficient pups, while there was also an interaction effect in the septum in which cells from male but not female iron deficient pups were larger. This may suggest that iron deficient cardiomyocytes were suffering from hypertrophy soon after birth, and it could be as a compensatory response for their hypoxic prenatal environment, in which their moms were severely anemic, and also to compensate for their own anemia soon after birth. It's important to note that cardiomyocytes stop proliferating around birth, so this hypertrophic compensation may have important long-term consequences. Next, in panels D, E, and F, we normalize the cross-sectional areas of the cardiomyocytes to their heart rates, and on postnatal day 28, we saw there's an significant overall increase in relative septal and right ventricular cell sizes in the iron deficient pups, and an interaction in which male but not female iron deficient pups had increased relative left ventricular cell sizes. Given that this is normalized to heart rate, this may indicate that there is decreased cardiomyocyte endowment at birth in the iron deficient pups, since again, cardiomyocytes are terminally differentiated at birth. And this could provide insight into some of the longer-term effects of perinatal iron deficiency, even after we saw that their iron status has recovered by day 28. So in conclusion, we saw that maternal iron restriction causes growth restriction and increases relative heart rate postnatally. We saw that the alpha to beta myosin heavy chain ratio was overall lower in the iron deficient pups on days 14 and 28, which may indicate delayed cardiomyosin maturation. Histological analysis showed evidence of neonatal cardiomyocyte hypertrophy, as well as decreased cardiomyocyte endowment at birth, which may contribute to the development of long-term cardiac dysfunction. And lastly, the remaining histological analysis into collagen deposition, as well as other measures, may provide insights into the mechanisms by which perinatal iron deficiency affects cardiac development, as well as the long-term cardiovascular health of the offspring. I'd like to thank all the sponsors of our labs, as well as all of our lab members, and thank you for listening.